Let's begin with the first presentation entitled Agonist of a cannabinoid-sensing GPR55 enhanced intestinal tight junction reassembly via AMPK and ERK-dependent mechanisms, presented by Ms. Nichapa Chindaduangratn. Please give a round of applause.
So, good afternoon, everyone. My name is Nichapa Chindaduangratn, currently a third-year medical student at Princess Phramongkutklao College of Medicine. Today, I'll discuss the effects of the cannabinoid-sensing G-protein coupled receptor 55 activation on intestinal tight junction reassembly and its underlying mechanisms. So, intestinal tight junction disruption is the main pathogenic feature of the inflammatory bowel disease, or IBD. Unfortunately, there is still a lack of FDA-approved drugs that directly targets these tight junctions. However, there has been increasing evidence supporting the use of cannabinoid in IBD treatment with unknown mechanisms, and therefore our main objective is to investigate the pharmacological activation of this cannabinoid-sensing G-protein coupled receptor 55, or GPR55, on intestinal tight junction reassembly. And T84 cells were used as our in vitro model. Firstly, the MTT assay was used to assess the toxicity of GPR55 activation by measuring the mitochondrial dehydrogenase activity. The transepithelial electrical resistance, or TER measurement assay, was then used to evaluate tight junction disruption in fully differentiated intestinal epithelial cells monolayer. Intestinal tight junction disruption was first induced using calcium depletion assay, and then the experiment was then carried out using the following groups. So control, 01602 with and without inhibitor of intracellular signaling. And since the tight junctions are the resistors of the cells, high TER indicates tight junction integrity and low TER indicates tight junction disruption. Finally, paracellular permeability assay was used to distinguish between intestinal leak pathway and apoptosis-dependent leak pathway. If tight junctions are disrupted, only 4 kilodaltons Fit-C dextran will be able to pass through, whereas in case of apoptosis, both 4 kilodaltons and 70 kilodaltons Fit-C dextran will be able to pass through. Our study has found that the molecule 01602, a synthetic agonist of the GPR55, was able to induce tight junction reassembly by suppressing intestinal leak pathway permeability without affecting intestinal viability. Figure A shows that there has been a drop in TER after the calcium switch, and so the treatment with 01602 mimics GPR55 activation and has yielded a significant increase in TER in a time and dose-dependent manner. This means that our treatment is effective because tight junctions would not have reassembled on their own even if we had switched back to normal culture media. Figure B shows the MTT assay, which illustrates that the molecule 01602 has no effects on cell viability, and so it means that GPR55 activation does not have any cytotoxic effects. However, the drop in TER does not always indicate tight junction leak, and so the permeability assay results must be considered, and the figure C and D shows that there has been a significant increase in 4 kilodaltons Fit-C dextran, but not for the 70 kilodaltons counterpart, which indicates that the drop in TER is the result of the tight junction leak rather than cell apoptosis. Finally, we also investigate into the effects of the GPR55 activation, which is the signaling cascade, and so the phospholipase C, calcium calmodulin kinase, AMP activated protein kinase, and extracellular signal regulated kinase and their inhibitors were tested. All 4 inhibitors have shown reduced effects of GPR55 activation, and so therefore we can conclude that the pharmacological activation of GPR55 induces intestinal tight junction reassembly through the PLC, CAMKK, AMPK and ERK signaling pathway as illustrated by this diagram. Finally, we hope that the findings of this study will constitute to a better understanding of the cannabinoids' effects on the human body, as well as pave the way for future developments of therapeutics targeting cannabinoid receptors. Thank you.
Your presentation really adds value to our conference. Any questions from our judges?
Thank you. Could you please explain the term Thai junction reassembly? What does it mean, and what is the process of this the process of this assembly?
Thank you for your question. So, the reassembly of intestinal tight junctions. So, these tight junctions they're able to reassemble on their own, meaning that they because in our study, for example, it was induced by the disruption was induced, so it broke apart, and so the reassembly by stimulating this receptor and by this pathway was able to reassemble them together, meaning that it conjoins the leak between the cells.
And normally, how long does it take for this reassemble? Is it needs also the new protein productions, or it just reassembled by the already existed unit?
So for in our study, we use so we cultured the cells for 10 days, obviously, and the tight junction disruption was induced for 16 hours, and so by this diagram shown, and so for the molecule after the induction of the tight junction reassembly by 01602, we left it on for like 2 hours, and and then the experiment was conducted after that. So the answer would be around 2 hours, because based on our study.
I just would like to make a comment, because the result you propose here is quite impressive, because the agonist can stimulate then change transepithelial resistance from only after first 30 minutes. So it's quite fast. So maybe some rigid pathway to reassemble. So this should be a good target.
Thank you.
For the figure B of the lower one over there.
The lower one?
Yellow one, yeah. Can you explain more about the statistical part, that which test that you used for the soil and the sign that you give here? For those figures, A to D.
Can you please repeat your question? I didn't
because
quite
you
catch
have
it.
the sign of the statistical significance in all the figure A to D. What are the tests that you use for each point? Because I understand that one way ANOVA should be tested for the whole group, but what is the test that you are mentioning here?
You mean the test for the this statistical test, right? I believe we use the one-way ANOVA, but we also have the aided help from our lab assistants as well, and so we didn't quite develop the statistical test on our own. Yeah.
Okay.
Thank you, Ms. Nichapa.
Now,